From the experiments that we have just conducted, we've gotten about 3.5 of the 5 I's of microbiology done. We inoculated our culture, we incubated it, we isolated it, and we microscopically inspected our unknown bacteria by viewing it under the microscope. Now that we've done those, we are ready to move on to the next 5 I's, which includes further continuation of inspection, but macroscopically, and doing the 5th I, which is identification. Before you can view the characteristics of our unknown bacteria, you will need to gather the right materials and set up your workspace. So the next experiment that we will be conducting involves using a sterile broth. So for this experiment, we'll need our unknown bacteria, which is right here. It is our slant stock culture. Our sterile broth, which is in this test tube right here. We will also need a solid nutrient agar for streak plating. We will need a frosted slide for our bacterial smear. We will need our inoculating loop. We will also need a China marker so that we can use it to label our plate and also create a streak pattern, but that'll be later on. And we also need a Bacto incinerator to sterilize our loop and to actually sterilize the mouth of our test tubes, which you'll learn later on. So the first thing that you would do before you begin is also make sure that you have on a new pair of gloves, goggles, and your lab coat. And now that you have all the materials needed, you are ready to begin your experiment. So to start off, you would label all of the media, slides, well, not media, but your plates, your slides, and your test tubes with your China marker. I've already gone and done that, as you can see in here and on these. And once you've put your initials on it, you are ready to begin the real experiment. To start, we are going to sterilize our inoculating loop by putting it in the Bacto incinerator for about 5 to 10 seconds. And then after we sterilize it, we're going to let it cool off. And just to speed up time, once the loop is cooled off, you're going to grab your slant culture, which is this right here, aseptically take the cap off, flame the tube for about 5 seconds. And once you've done that, you're going to take your sterilized inoculating loop and you're going to scrape a bit of the culture from the slant, flame the tube for 5 seconds, put the cap back on, and then put the test tube back on the test tube rack. Now what we're going to do is we're going to grab our broth, take the cap off, flame the tube for 5 seconds to sterilize the opening, and then we're going to take our inoculating loop that has our unknown bacteria on it, and we're going to stick it in the broth. We're just going to gently move it back and forth so that the bacteria gets distributed through the broth. Once you've done that, pull the inoculating loop back out, flame the test tube for 5 seconds, put the cap back on, and then put the broth test tube back on the test tube rack. And then we're going to sterilize our inoculating loop for 10 seconds in the Bacto incinerator. Once you've done that, set your inoculating loop on your benchtop. Now what you're going to do is you're going to put your culture that has the broth and your unknown bacteria in it in the incubator, and you're going to incubate this culture for 48 hours at 37 degrees Celsius. After you've put your culture in the incubator, you have to disinfect yourself and your workspace. You would first move all the materials out of the way. You would clean it by using germicide. Well, not clean it, but disinfect it with germicide. And then use paper towels to wipe the surface down and also to dry off your workspace. Then throw away any of the used paper towels in the biohazardous waste container. Once you've done that, remove your gloves and put them in the biohazardous waste container as well. Then go wash your hands thoroughly though.